drill down a little farther into the epithelium and the junctions which we find predominantly in the epithelial cells. The first set of junctions we're going to talk about, in fact, are the hemidesmosomes, but we're going to subsume here those hemidesmosomes under a broader category called anchoring junctions. And I think you can infer from what we talked about on Monday that these anchoring junctions are going to hold those cells fast to the basal lamina. We're going to walk our way through the hemidesmosomes. We're going to talk about a few of the proteins that are in those hemidesmosomes. We're just going to skate by some of the major proteins. If you get down into the weeds of this, and if you like rabbit holes, you can go down this rabbit hole. That we know a whole series of proteins that are part of these hemidesmosomes. We know how they interact with one another. We know how they self-assemble. But I just want to give you the basics on that and have you hold on to the idea that it's the hemidesmosomes and a complex of proteins, particularly with the integrins, which are transmembrane proteins, that are going to hold the cells fast to the underlying basal lamina. And the basal lamina, again, is extracellular proteins that have been secreted in part by the epithelial cells and probably in part by underlying cells, mostly fibroblasts, that we'll talk about later. But another type of anchoring junction is the so-called desmosome. As the name implies, a hemidesmosome is half of a desmosome. And it's a little bit of an exaggeration. There are different proteins in them, different molecules. But when these were first described from the electron microscopy, they really didn't see the distinction because they didn't know what proteins were in them. They called one half a desmosome and the other desmosome. For our purposes, what we're going to see is the integrins are part of the hemidesmosome and they anchor cells to the underlying basal lamina, whereas desmosomes are cell-cell interactions. Desmosomes hold cells fast to one another, and they use a slightly different set of proteins. They'll, in fact, use a series of proteins called cadherins, and we'll talk a little bit about them. Then the next set of junctions we're going to talk about are the so-called occluding junctions. The occluding junctions are commonly known as tight junctions. And again, this term tight junctions goes all the way back to the old 1960s or so, maybe a little earlier, when they were first spotted in electron microscopy. And they were named tight junctions because the cells came close together and appeared not to have any space between them. The membranes were tightly adhered. That's where the name comes from. We're going to talk about them in terms of occluding junctions. We're going to see that these zipper cells together. Now, it's a bit of an exaggeration to say they zipper the cells together in that there's not much strength in these junctions. The real strength in holding cells together is in the desmosomes, and we'll go through the protein complexes that make these junctions so strong. The occluding junctions, the tight junctions, are not particularly strong. They're they're just basically a few proteins in the membrane that are holding two cells together. Their function is not to hold the cells together. The function is to provide a kind of dam, a kind of occlusion, a kind of position where we can't get small molecules through between adjacent cells. So we'll draw some of these and talk a little bit about the molecules in them. And then the third type of junction we're going to talk about are the communicating junctions. And these are the ones that I think are the most important that we're going to refer to more as we go on to the course. The communicating junctions have another name. They're called gap junctions. And again, this name derives from the early microscopy where they could see in electron micrographs that the cells came close together but didn't quite touch. Unlike the tight junctions where the membranes appeared to touch one another, the gap junctions had a case where the two membranes were clearly separate from one another, separated about 20 nanometers. But the important idea is that these junctions, these gaps, are bridged by proteins. There's going to be a protein bridge between the two cells, and not only are the cells bridged by proteins, but these cells now will form channels. So now we have cytoplasmic continuity between adjacent cells through these gap junction channels. The gap junction channels have a name. They're called connexons. They are formed from 6 smaller proteins called connexins. The 6 connexins form a connexon. That forms a channel between the two cells. Each of the two cells will have a set of connexons which associate in the extracellular environment, providing a hydrophilic pathway. Once we have talked about the junctions, we're then going to move on and talk a little bit more about a specialization of epithelial cells. The epithelial cells we've talked about so far have been pretty much nondescript blue boxes that we placed on a basal lamina. And this is going to be one of the first places we're going to find that epithelial cells themselves can generate a kind of function, and these are going to be the so-called exocrine glands. The exocrine glands have ducts. Unlike the endocrine glands we'll talk about later this week or into next week, these actually have ducts that go to the outside world. Among the ducted glands are the sweat glands, the salivary glands, and the mammary glands, and I'll draw some of these. And what we'll see in common for all of them is that there's a surface epithelium to which the duct is connected. The duct is composed of a series of cells which we call a duct epithelium, and that then extends into a secretory epithelium. It's the secretory epithelium which is involved in secretion of some molecules, but in some cases just generating a gradient. We'll talk a little bit about that. But basically we have a secretory epithelium that's continuous with a duct epithelium that extends out of the surface, forming a surface epithelium. And in this way, milk from mammary glands, saliva from salivary glands, and sweat from sweat glands can go out onto the surface. Then we'll begin talking a little bit about connective tissue. Connective tissue is divided into 4 types. There's connective tissue proper, cartilage, bone, and blood. And we'll drill down into each one of these to some extent. Most of the story in connective tissue is going to be told in the form of skin, whereas connective tissue proper is going to be the dermis of skin. We'll talk a little bit about cartilage, emphasizing that it's a true tissue. It's only composed of one cell type. Then we'll talk a little bit about bone, and bone is in fact an organ. Bone is in fact composed of the connective tissue type of osteocytes, but in addition to that, it will be innervated and it will contain a lot of connective tissue proper. So it's in fact a proper organ. Then finally, we'll talk about blood, and we'll talk about the cells of origin of all of these. But when you think about a connective tissue, what I want you to think about, first principles of connective tissue, is that they're individual cells, but they're separate cells that are embedded in a matrix they have secreted. That's in contrast to the epithelium, where the epithelium are these broad sheets of cells, all stacked one next to the other. Here in a connective tissue, whether it's blood, bone, cartilage, or connective tissue proper, we have a cell type, which will be hematopoietic stem cells, or osteocytes, or chondrocytes, or fibrocytes. But what's characteristic of all of them is that these cells are usually separated from one another, and they're embedded in a protein matrix that they themselves have secreted. And we'll get into all of these later on in the course. But let's begin where we left off and begin talking about hemidesmosomes. As anchoring junctions. Again, let's imagine we have an epithelium, and it's a simple epithelium with these epithelial cells sitting one next to another. And again, remind you that if this were a proper epithelium of cells, it would come out into the audience and behind the plane of the blackboard. And that as an epithelium, it would be attached. It would be sitting on an extracellular series of proteins called a basal lamina. Now again, the basal lamina are a whole host of different extracellular proteins. And what we're going to drill down into now is to talk a little bit more in detail about where we finished yesterday, or Monday, where we have one of these epithelial cells. This is the apical surface. This is the basilar surface. Remember, we said that a characteristic of all epithelial cells is they're polarized, and that as such, they have an apical and a basilar surface. And here, and I'll put this in orange, There are a whole series of proteins down here, and I'm not going to tell you about them because they're a whole flock of them. And I'm just going to label this as a protein plaque. We know all of the proteins that are in the protein plaque at this point. We know that they're all connected one to another. But we also know, more importantly, we've known this for a long time, that that protein plaque is connected into cytoplasmic proteins. Now, in skin, these proteins are keratin. In most cells, these cytoplasmic filaments are actin. But regardless of whether we're dealing with actin or we're dealing with keratin, we have these cytoplasmic polymers of proteins, and they connect into this protein plaque. Now, there are specific interactions in here we're not going to get into, but what I want to emphasize here is that in black, there's a protein that comes across the membrane. And this protein is said to be integrin. If you get into the weeds of this, you'll find it's a dimer, it's a beta 4 alpha 6. In some cases, there'll be a beta 1 alpha 6. Don't worry about that. These are integrins, and these are transmembrane proteins. These are transmembrane proteins that, if you think about this as having their feet are embedded in the protein plaque. They extend across the phospholipid bilayer, so they're going to have alpha helices here across the phospholipid bilayer. And out here in the extracellular space, they're now connected to other proteins. One of the proteins that we commonly see connected here is called laminin. And this laminin then is further connected into proteins like collagen. Get into the details, it's collagen 7. All of these proteins have names. What I want you to recognize is that we have this complex, that the major player that I want you to remember is integrin. Integrin is a transmembrane protein that links the series of proteins in the protein plaque and the cytoplasmic filaments to the extracellular proteins in the basal lamina. So this is holding the cell fast to the basal lamina. Now, just to point out the importance of this, there is a disease. I have to read this, sorry. There's a disease called bullous pemphigoid. Bullous pemphigoid is a disease which is an autoimmune disease. And what happens is that one of the components of this transmembrane region of the integrin is attacked by the immune system. And it's a horrible disease to get, because what happens is these epithelial cells, particularly in the skin, will now come loose from the underlying basal lamina, and it's a blistering disease. So what happens with people with extreme forms of this disease is the slightest bit of shear on the skin, which is an epithelium, will tear it loose from the underlying basal lamina, and a blister will form. And this is because some of these transmembrane proteins of parts of the integrins are across the membrane with their feet in the plaque, tear loose and disrupt the ability of these epithelial cells to be anchored to the underlying basal lamina, just pointing out the importance of these anchoring junctions. But let's go to the right-hand panel and talk about the next kind of junction. This is going to be an anchoring junction. And this anchoring junction is called a desmosome. And this anchoring junction anchors cells to one another. So that if we look at one of these epithelial cells and its neighbor, let me put those a little farther apart. Now, where it's true, these cells will be anchored to the underlying basal lamina by hemidesmosomes. Where these individual cells will be anchored by hemidesmosomes to the basal lamina. Through the integrins. They, in addition, will be anchored to one another by transmembrane proteins up here, called cadherins. Again, if you look this up and you get down into the weeds, you're going to see that these cadherins have their own names. And we're not going to get into that. We're just going to call them cadherins. And the cadherins are transmembrane proteins that link cells one to another at desmosomes. You could easily say, rather than link, you could say anchor. Well, just as we saw with the hemidesmosomes, there's going to be a set of proteins. These are different proteins, but they are proteins in the cytoplasm here. And again, all of these are named. If you want to look the names up, that'd be fine. But I'm just going to say this is a protein plaque. And then connected to that protein plaque, again, will be either actin in most cells, or in epithelial cells, of skin, these will be keratin filaments. And this gives rise to this cell type we're going to talk about a little later, called keratinocytes. We'll see the keratinocytes are the epithelial cells in skin. So those are the anchoring junctions. These are what hold cells one to another, sometimes, and taken together. This is said to be a desmosome. And in some cells, you'll see desmosomes that go all the way around the cell and will anchor all the cells to its neighbors. This is a fairly strong junction. This is a pretty mechanically strong junction because these protein plaques are pretty stout, pretty robust. They're connected to the cytoplasmic filaments, and then the cells are being held together by these cadherins in the extracellular space. So, are there any questions about anchoring junctions? Okay, so if there are no questions about anchoring junctions, these are holdfasts. They hold cells to one another. They hold cells to the underlying basal lamina. And over here, I want to now talk about a different kind of junction called a tight junction. Before we leave these, let me add one more point of interest, because I think as time goes on, we're going to find that this is going to be more and more important to people in their lives. And that is one of the characteristics of an epithelium is that the cells divide, cells replicate. So all epithelial cells maintain this ability to replicate, and if you think about it for a minute, that's pretty important. I mean, if this morning you had a piece of toast and you scraped the roof of your mouth, you're not too worried about it, because those epithelial cells will in fact replace themselves. They'll basically divide and heal and make new copies of themselves. Well, this is true of all epithelia. The problem is that every time one of these epithelial cells replicates, there's a small but finite possibility that it becomes a cancer cell. And this cancer cell now no longer adheres properly to the basal lamina. It's said that the cancer cells lose their respect for the basal lamina. They go wandering off and they start setting up housekeeping in other places. And that's when you have a metastatic disease. It's really dangerous, because these cells start dividing out of control in places where they don't belong. And in fact, if you have ever read a pathologist's report about a tumor, you'll often see them referred to as carcinomas. Whenever you see this carcinoma, this tells you that the tumor, the cell of origin for the tumor, was an epithelial cell. Could have come from the skin, could have come from the lung, it could have come from any number of places. But if it's a carcinoma, its cell of origin was an epithelium. In fact, in some cases, we'll see something known as an adenocarcinoma. Give myself some room. An adeno An adenocarcinoma is a cancer, a tumor, that originated as an epithelial cell that was part of a gland. And we'll get into that before the end of today, talking about epithelial cells as glandular structures. So any questions on that? Okay. Then let's begin talking about tight junctions. Tight junctions are also known as occluding junctions. And if we use our terminology here of two blue cells being epithelial cells, well, we've changed the shape of the epithelial cell a little bit here. And there are places where the two plasma membranes approach one another. And if we look carefully at these sites of close apposition, we think that these membranes actually touch one another. That's why they got the name tight junction, but they don't. They're separated by a little bit, but there are in fact proteins in here. Now, these proteins, I'll name a few of them. Some of them are called JAM, some of them are called occludins, some are called claudins. Don't worry too much about that. Just know that there are proteins here that are meeting up in the extracellular space. And by meeting up in the extracellular space, they provide a block for the movement of small molecules between cells. Now, let's see if I still have some in here. You can imagine two cells being attached one to another. See if I can make these work. Oh, here's one right here. So imagine these are the two cells. These two plastic bags, these two Ziploc bags. This cell, this plastic bag is over here. This plastic bag is over here, these two cells. And they have now been zipped together by the Ziploc. That's pretty much like these proteins, these Jam, Macludin, and Cloudin proteins, which are zipping the two cells together. Now, they're not very strong. We can pull them asunder fairly easily, but what they do is they provide a blockage. They prevent the movement of molecules from the apical surface down between the cells toward the basilar surface. Don't forget that if we were to think of these cells as epithelial cells, of the gut. If we imagine these as epithelial cells lining the stomach, and this is the lumen of the stomach, the contents out here are pretty corrosive. The pH can be down around 2. It's full of proteolytic enzymes. You don't want the muck that's up here in your stomach to be coming down between the cells and entering the tissues below. So we zipper these cells together with the tight junctions, and we occlude the space between them, preventing the movement of small molecules between cells down into the underlying tissues. That's the function of a tight junction. Let me show you a couple of slides here, and then I'll finish with a story of the gap junctions. The first slide is intended just to show the epithelia. This is a simple epithelium. This is meant to be the epithelial cell here. This is the nucleus sitting on a basal lamina. This is a stratified epithelium where we have these cells stacked. We'll talk about that a little later when we talk about skin. This is a very schematic, highly stylized description of anchoring junctions. The one down here shows the protein plaque linked into the keratin filaments. Again, we know that this is a complex of proteins down here, and this complex of proteins is then connected between the cytoplasmic proteins and the basal lamina, and that connector is through this structure here, the integrin. Now, we blithely show this is a block of proteins and show this is a transmembrane protein here. But in fact, there are probably any number of control mechanisms in here, and cancer biologists are really interested in these because they would like to know what goes wrong in these plaques that are normally anchoring the cells to the basal lamina when these cells leave their attachment to the basal lamina, wander off and live someplace else. So these regions, the proteins in the hemidesmosomes as well as in the desmosomes, the cytoplasmic side of the protein, the regulation of these proteins is becoming an important discussion point in cancer biology because we'd like to know what these control mechanisms are in terms of keeping these cells where they're supposed to be and not letting them wander off and grow up where they're not supposed to be. But again, here's the integrin, here's the protein plaque connection to the cytoplasmic filaments, which could be keratin and keratinocytes or actin in other cells. We also said that these cells are anchored one to another, and these potato masher-like structures are meant to be the cadherin molecules. They're in the extracellular space. They're linked together in the extracellular space. You can think of these almost like Velcro, the hook and eye on Velcro, where these proteins are linking one to another, and they're being held fast across the membrane. Because again, these cadherins are transmembrane proteins. They're being held fast because the cadherins are linked to a series of proteins in this protein mat. And these are further connected to the cytoplasmic proteins of keratin and keratinocytes and actin in most other cells. This is an image of a tight junction. This takes a little bit of explaining, but if you've been following along the structure of phospholipid bilayers, I think it's not so bad. They're showing here the two leaflets of the phospholipid bilayer. This is the leaflet that's closest to the cytoplasm. So this would be the cytoplasm up here of this cell. They've peeled back the leaflet of the phospholipid bilayer that's closest to the cytoplasm and revealed the leaflet of the phospholipid bilayer that's closest to the outside world. Then they're showing these beads in here and these sort of complicated paths of beads, and these are meant to be the proteins of the tight junctions. These are meant to be occludins and claudins. And so these are in the phospholipid bilayer, and then they extend into the extracellular space. And in the extracellular space, the claudins and occludins now are linking one to another, and they're linking these cells together. But unlike the desmosomes, which are spot welds, these are continuous zippered together structures, so they prevent the movement of molecules between the cells. So again, this is cytoplasmic leaflet, this is the extracellular leaflet, this is the extracellular leaflet of the neighboring cell, and the cytoplasmic leaflet of the neighboring cell. And these are the proteins embedded as transmembrane proteins in the two cells that are forming that tight junction. Any questions on that?
Can you just briefly reiterate the role of the keratin filaments?
What's the role of the keratin filaments? The keratin filaments are cytoplasmic filaments. These are intermediate filaments. So they're polymers of intermediate filament protein. They then are connected to the protein plaque. You can almost think about them as a root anchoring the cell to the underlying basal lamina. And these are the root, and then you have the part of the plant that goes to the outside world through the plasma membrane. So these provide tensile strength to hold this plaque together and to hold the plaque to the underlying basal lamina through the transmembrane protein of the integrin. And in most in cells of skin, this is keratin. In most other cells, this will be actin. Anything else? Okay. Well, let me then go on and talk about take this middle panel again and describe another kind of junction. And this kind of junction is said to be a gap junction. And it gets its name, again, from the early electron microscopists, who saw that there were these places where cells came really, really close together. But they didn't quite touch. They contained a gap between them. And because there was a gap between the cells, in contrast to what they saw with the tight junctions, they said there's a space or a gap between the cells, so they called them a gap junction. They really didn't know what their function was. They didn't know what they were made of. We now know that the gap junctions are the sites where transmembrane proteins reach across the extracellular space and form intercellular channels between adjacent cells. And so we'll see then that these gap junction proteins are going to provide a kind of hydrophilic pathway, a kind of channel between cells. We've talked about channels that go across phospholipid bilayers, but now we're going to talk about channels that not only go across the phospholipid bilayer, but in addition, link the two cells together, providing a kind of cytoplasmic continuity. So let's draw one of these epithelial cells. Again, I'll leave a gap here. Again, when the microscopists first saw this, they measured it, and they said this is a 20 nanometer gap. They really couldn't measure the gap in the tight junctions, so they called them tight junctions, and they said there was a gap in here. And then they started to do experiments, and they found that there were proteins that spanned the gap. And with further purification, they found that the proteins that spanned the gap were called connexins. And they further found that 6 connexins formed a connexon. And the connexon is the channel. So I'm going to show you this a couple of parallel rows and talk about how we have cytoplasmic continuity, and then we'll drill down a little farther and see how this happens. So there's this is a transmembrane protein, and transmembrane protein on this side, and they meet up in the extracellular space. But now there's a channel. This is an intercellular channel. This is an intercellular. An intercellular hydrophilic. Pathway. Now, in this typical gap junction, there may be, tens or hundreds of these transmembrane proteins, these connexons. But what I want to do now is I want to imagine two ways of thinking about this. You can imagine sticking your head inside the board and looking down on this. Or we can imagine taking this region of the membrane, turning it at right angles, and looking down on it. And when we do that, as we talked about before, we see the phosphate head groups. These are meant to be the phosphate head groups of the phospholipid bilayer. So phosphate head groups. Of phospholipids. But then there are transmembrane proteins here. And I'll put these in red. Each one of these red circles is meant to represent something called a connexin. C-O-N-N-E-X-I-N. That's the protein. There are 6 connexins form a connexon. So this structure here, formed from the 6 connexins, is a connexon. Each of the connexins has, each connexin has 4 transmembrane alpha helices. So again, if you go back to when we talked about how we'd form a channel, there will be 4 alpha helices in each one of these. Those spiral staircases going down across the phospholipid bilayer. And they will have hydrophilic, polar and hydrophilic R groups pointing inward. So now these polar and hydrophilic R groups provide a hydrophilic pathway across the phospholipid bilayer. But in addition, this connexin connects with this connexin in the extracellular space to form a channel between the two cells. So now we've generated a hydrophilic pathway that connects the two cells one to another, providing cytoplasmic continuity. It turns out molecular weight molecules of less than about 1,000 can pass through these gap junctions. So that means ions freely move through. Things like ATP freely move through. Once you begin to get to things the size of ribosomes, they're too big. But things of molecular weight of about 1,000 can pass freely through these. So are there any questions about the structure of these gap junctions? Let me show you one picture then. To try to bring that all together, and then we'll talk about glands. So again, here are 2 cells. There's one cell up here and one cell down here. These structures here are meant to represent the 2 leaflets of the phospholipid bilayer. This is the leaflet of the phospholipid bilayer closest to the cytoplasm of this cell. This is the leaflet of the phospholipid bilayer closest to the extracellular environment. This is the leaflet of this cell closest to the extracellular environment, and this is the leaflet closest to the cytoplasm. Then shown here, sort of opened up, are each connects on. So here's the 6 connects ins, 1, 2, 3, 4, 5, 6. We've opened that up, and this shows the channel that extends between this cell and this cell. So this is an open channel between cells. This will all be hydrophilic, or polar at least, amino acids in here, providing a hydrophilic pathway. If we look at it in this dimension, we see the 6 connects ins going around the channel. There's the channel. You can imagine things moving back and forth between this cell and this cell, where they're pasted together here between this cell and this cell, where these guys are pasted together in the extracellular space. This shows just the connects on in this cell. This shows connects on in this cell, and they're trying to show here where the connects on from this cell is matched up with the connects on from this cell, forming that hydrophilic pathway between this cytoplasm and that cytoplasm. Is there any question on that topology?
Would you mind just reminding us all the difference between the terms intracellular and intercellular?
Oh,
When we talk
okay.
about these channels?
Okay, when we talk about intercellular, means between cells. So we now have communication between cells. Intracellular means within the cell. So intracellular is contained within the cell itself. So intracellular transport was like that cytoplasmic transport. Intercellular is between cells. So inter between, intra within. Anything else? Right. Well then, talked about junctions. Let's now move on a little bit and talk about specializations of epithelial cells. And I'm going to label this panel as epithelial cells as exocrine glands. And this does two things. It first describes what an exocrine gland is, and it shows how the epithelial cells in an exocrine gland are organized and oriented. Now, the exocrine glands will have a name. They're called ducted glands, and we'll show this in our image. And they connect to the outside world, so they connect to the surface. So they're going to have a duct that goes out into the surface, and then they're going to have a secretory epithelium, which is below the surface. And the secretory epithelium is going to be generating the secretion, which is then going to go to the outside world through the duct. So let's first describe these as exocrine glands and say that exocrine glands are made of epithelial cells. And the exocrine glands include sweat glands, salivary glands, and mammary glands. Now, of course, these are making different things. Sweat glands are making sweat, and salivary glands are pretty closely related. They're making saliva. Mammary glands are more complicated because, after all, these are glands that are unique to mammals. Mammals are characterized by having mammary glands where the female of the species generates milk to suckle the young. And it's these mammary glands, these exocrine glands that are generating the milk. So they contain a little bit more of a machinery to make the milk. But by and large, they have these similarities in them in that they're ducted glands. And what we mean by duct is that there will be a series of epithelial cells, and I'll show you these by these sort of individual ovals. So this is an epithelial cell. These epithelial cells, as all epithelial cells must be, are anchored by hemidesipsomes to a basal lamina. So there's the basal lamina. So far all we have shown is a simple epithelium. But these epithelial cells will extend into another series of epithelial cells which are a little more highly specialized. And these are said to be the duct epithelium, or epithelial cells. They too are attached to a basal lamina. And then down here, at the bottom of this structure, are another set of epithelial cells, and we name them slightly differently. We say these are the secretory epithelial cells. They too sit on a basal lamina, and all of these cells have slightly different functions. So let's take the example of a salivary gland. What will happen with the salivary gland is these secretory epithelial cells are now going to be pumping sodium chloride across this membrane. That generates an osmotic gradient so that water will follow. Water follows and moves into the duct of the epithelial cell. Then the sodium and chloride, which has been used to draw the water, that sodium and chloride is largely removed from the duct epithelium and is predominantly replaced with potassium and bicarbonate. It's the job of the duct epithelium here in the salivary glands, and it's true as well in the sweat glands, to pump out the sodium chloride, replace it with potassium bicarbonate, and allow this dilute potassium bicarbonate solution to go out onto the surface. Now, if it's a salivary gland, the surface is going to be in your mouth, and you generate saliva. If this were out on your skin, this would be a sweat gland, and this would now allow for sweat to go out onto the surface. The secretory epithelium down here for the mammary glands is more complex because, after all, it's got to be moving fat and protein and all manner of large molecules. So this is a pretty complex structure. What's more, it's under hormonal control. It's under hormonal control of prolactin. Its development is under hormonal control of estrogens and progesterone. So this is a far more complex set of epithelial cells, but the idea is the same. And then we have a secretory epithelium, a duct epithelium, and then a surface epithelium. And sometimes if you read a pathology report of a mammary tumor, they'll talk about a ductal carcinoma, and that's a carcinoma that's been generated by the duct cells. If it's an adenocarcinoma, then it's a tumor that's been generated initially by problems with the cell division in these epithelial cells forming the secretory epithelium. And again, because they're under hormonal control, that various hormones will have an effect on the rate of division of those cells. And that's one of the major ways we control mammary tumors, is we find what kinds of hormonal control they're under and try to block those hormones. Well, we've gone beyond 50 minutes. Let's end it here, and we'll come back to this story next time.